Top 10 things you need to know about mitosis is used to mean exact copying of cells, division of nucleus. Why is mitosis important? It helps from babies and restores skin or a tail on a lizard. Idea of diploids versus haploids. Haploids has a copy of all of its DNA. In eukaryote cells, our DNA is organized into chromosomes or linear stretches of DNA, aka N, has N copies of chromosomes. Diploids basically has 2 copies of haploids, aka 2N. Cell cycle is basically how we go from one cell. Once we start, we continue to make copies after copy. Eventually, it makes 2 cells. It makes copies after copies. G1. A cell grows, called interphase. S phase is a part of G1. It's where the cell stops growing and makes it copies of the DNA. 6. G2 has all the phases. The cell continues to grow. After all these phases, the cells are ready to divide. It goes through mitosis. 1. Phase, metaphase, prophase, metaphase, anaphase, telophase, and cytokinesis. It's mostly spent in the interphase. The brain and muscles are not going to make copies. 7. Chromosomes is a X shape. One cell is chromatid and the other is sister chromatid. Mitosis is divided into 5 phases. A. Inner phase. The DNA in the cell is copied in preparation for cell division. This results into 2 identical full sets of chromosomes. 2. Outside of the nucleus are 2 centrosomes, each containing a pair of centrioles. These structures are critical for the process of cell division. 3. During interphase, microtubes extend from these centrosomes. Now B. Prophase. The chromosomes condense into X-shaped structures that can be easily seen under a microscope. Each chromosome is composed of 2 sister chromatids containing identical genetic information. The chromosomes pair up so that both copies of chromosome 1 are together, both copies of 2 are together, and so on. At the end of the prophase, the membrane around the nucleus is the cell dissolve away, releasing the chromosomes. The meiotic spindle, consisting of the microtubes and other proteins, extends across the cell between the centrioles as they move to opposite poles of the cell. Metaphase. The chromosomes line up neatly end to end along the central equator of the cell. The centrioles are now at opposite poles of the cell with meiotic spindle fibers extending from them. The meiotic spindle fibers attach to each of the sister chromatids. D. Anaphase. The sister chromatids are then pulled apart by the meiotic spindle, which pulls one chromatic to one pole and the other chromatic to the opposite pole. Telophase. At each pole of the cell, a full set of chromosomes gather together. A membrane forms around each of the set of chromosomes to create 2 new nuclei. The single cell then pinches in the middle to form 2 separate daughter cells, each containing a full set of chromosomes within a nucleus. This process is known as cytokinesis. Onion root cells. Roots grow by making more copies of mitochondria, but not least cancer. It is cells reproducing out of control. Cells have all of the mechanisms inside that stop cells from doing so. There will be a mutation, and it will suppress some of these cancer-suppressing genes and will eventually cause cancer. It can occur anywhere in the body and is able to activate other cells to become cancer called a tumor.